Okay, so in addition to inoculating that CNA blood agar plate today, I'm also going to have you do a gram stain from that broth culture, that mixed broth culture. So it'll be a good opportunity to practice doing a gram stain from a broth because you're going to need to do that later on when you do your unknown. Okay, so in addition to gram staining and inoculating that selective media, you have 3 more things to inoculate. And the Catalase test is a really nice, easy test. I love it because you don't even need to incubate it. So you get your results back right away today. Okay, and this is a really important test because it's one that differentiates our Staphylococcus from our Streptococcus. Of course, those 2 organisms can look pretty similar when we gram stain them because they're both gram-positive cocci. All right, so the Catalase test is one that we're going to actually inoculate on a glass slide. But let's talk about what the catalase enzyme does. Okay, this is an enzyme that breaks down hydrogen peroxide into water and oxygen. 1, 2, 3, 4, balance that out. Okay, and this is a really crucial enzyme because hydrogen peroxide is a byproduct of aerobic respiration. So any organism that respires aerobically needs to have a way to detoxify this hydrogen peroxide. Let's call it a toxic byproduct of aerobic respiration. Okay, so organisms that respire aerobically have to have some way to detoxify that. There are a couple different enzymes that do that. The Staphylococcus have catalase. Streptococcus have a different enzyme that we'll talk about next time. Okay, so the way you inoculate this today, since we have 3 different staffs, we want to test the catalase for those 3 staffs, and we're going to compare that result to how Streptococcus acts in this test. So today we're going to inoculate 4 different organisms. If you only wanted to do a catalase test for 1 organism, then you would just put 1 organism on your slide. But today, since we have 4 different things, you can maybe split that into quadrants. You can number it if you think you might get mixed up. With your loop, you are going to take some organism from, you're going to want to take this from some solid media so you get enough organism. So with your loop, get enough organism and you're just going to wipe it on that glass microscope slide dry. Okay, you don't want to add a drop of water on there. That's going to dilute the results. So it seems kind of weird just to take a colony and streak it on there dry, but that's what we're going to do today. Okay, so flame your loop, get your second organism, say maybe this was Staph epidermidis, and you're going to get some Staph aureus or whatever order you decide to do it. Rub that on there, get your Staph saprophyticus. Make sure you're flaming in between here, letting your loop cool in between. And then you're going to put, I think it's strep pyogenes that we have for you today. So put all those organisms on there, and then all at once you can go through and add a drop of hydrogen peroxide. We're going to have some 3% hydrogen peroxide for you in the lab there. Just put a drop on each one of those smears of cells there. Okay, and what you're looking for a positive result is some vigorous bubbling. Okay, you are looking for the product of the reaction that we can see. Okay, and that's the oxygen bubbles. Okay, negative results, you might get a few bubbles, let's say few or no bubbles. Okay. You probably want to set this glass slide on the black bench top here. If you're setting it on top of your lab book with a white background, it's just a little bit harder to see those bubbles. So put that right down on the bench and convince yourself that the 3 staphs are vigorously bubbling and the strep is not bubbling almost any at all, maybe a couple bubbles. Okay, so convince yourself that those look different. If they don't look different, try it again. Maybe try a different bottle of hydrogen peroxide. That does tend to go off, and we haven't had problems with that for a long time, but that's why the hydrogen peroxide is in those brown glass bottles, because eventually the light basically disassociates it into water. Okay, so if the hydrogen peroxide is not good, then this isn't going to work, and just convince yourself that that works. I should also mention, make sure that you don't get too overactive, I'll say, and when you're taking some cells from your solid media, you don't want to dig in to that, especially if it's blood agar, and get any media on your slide here, because blood agar actually itself tests positive for catalase, okay, because there's blood in there. So just make sure you don't get too heavy-handed there and that you're only getting cells. This is why we have to do the test on a glass slide, and we can't just drop the hydrogen peroxide right on the media, because some of the media will start to bubble. Okay, so we make catalase in our cells as well. Back in the day, we used to use hydrogen peroxide as an antiseptic. Say if I'd fall and skin my knee, Mom would put hydrogen peroxide on there and it would bubble a whole bunch. That's not used as much anymore as an antiseptic, because it's really fine to use it, when you first fall to clean that wound out, but you don't want to keep reapplying hydrogen peroxide every day to clean a wound, because actually we found that it inhibits our cells from healing from that wound. So, it's not good for our cells. So using it once, good, but I guess we're more often use other things now. Okay, lots and lots of organisms produce catalase. Okay, it's not just Staphylococcus. It's an easy way to tell Bacillus, an aerobic genera, apart from Clostridium, an anaerobic one. Almost all of our gram-negatives that we use in lab are catalase positive. So again, lots of organisms catalase positive. It's an important test for us to tell the difference between staph and strep. Okay, so once we know we have a staph, really it only takes 2 tests to tell the difference between our 3 staphs. And the coagulase test, this is another enzyme. It kind of sounds like catalase, so sometimes students are a little bit confused. We're not going to talk too specifically about the actual mechanism of this enzyme. I'm going to summarize what it does. It basically, in the blood, converts fibrinogen to fibrin. Okay, the actual reaction is pretty complex, and I'll read this to you, but don't write this down or memorize it. Coagulase reacts with prothrombin in the blood, which results in staphylothrombin, which enables the enzyme protease to convert fibrinogen to fibrin. Okay, the result here, what we're interested in, is that it forms a clot in the plasma or in the blood. Okay, this is actually an exoenzyme. And we skipped our whole lab on extracellular or exoenzymes, but that just means it's an enzyme that bacteria secrete outside of their cell. Okay, so it does all its work outside of the cell. So what good is it for an organism to produce coagulase? Why would it want to clot the blood? Well, let me draw a little picture here. Say this is the skin, and say somebody has gotten a little cut in their skin, And these little dots are some bacteria that have started to multiply in that cut. All right, skin is a really great protective barrier, but when it gets cuts in it, then obviously that's a great portal of entry for bacteria. Okay, if those bacteria can secrete coagulase, okay, that enables them to kind of clot up the blood around that site of entry. So it sort of enables them to kind of wall off themselves, we'll say, okay, from our host kind of, we'll call it non-specific immunity. So things like white blood cells, like neutrophils or macrophages that would come and engulf those bacteria, okay, those white blood cells now can't get to this site of infection because they've secreted coagulase and kind of walled themselves off. Okay, so organisms that produce coagulase, it makes them more likely to establish an infection. Okay. Okay, so we would call it a virulence factor. It helps the organism, be successful in causing an infection. Okay, so this is not, the secretion of this enzyme is not common in the microbial world. One of our Staphylococcus does it, and hint, it's the one that caused all the different kinds of infections. The organism Yersinia pestis makes coagulase. That's the organism that causes bubonic plague. So it's sort of here and there in the microbial world, but not too common. Okay, to inoculate this, you are going to use a kind of smallish test tube that has a little piece of foil on the top of it instead of a cap. These are supposed to be disposable test tubes that we clean out and use over and over again. There's a small volume of rabbit plasma in there. This is one of our more expensive things that we inoculate. So if you goof this up, just ask me before you get a new one. Okay, so you're going to take that foil off and just inoculate this. Not quite exactly like you would a normal broth. You want to get a pretty heavy inoculum. So what does that mean? That means when you're picking up a colony from the plate, get enough that you can see on your loop, and when you stir it around in that rabbit plasma, it should look a little bit cloudy. Okay, convince yourself that that rabbit plasma is totally liquid when you inoculate it. Okay, your book says that it only has to be incubated for 4 hours. We usually leave it overnight. And then for a positive coagulase test, what we're looking for is for that rabbit plasma to clot. Okay, and usually the whole thing is solidified. Usually you turn it upside down and it's totally solid. Sometimes you'll just get a chunk or two. That's enough to call it a positive. Okay, negative result is that it's still liquid. Okay, you want to make sure that you put that foil back on after you inoculate it, because there's so little volume here that a lot of it would evaporate away, or if not all of it, in the incubator. So make sure that that foil is back on there to keep that from evaporating. Okay, and again, as long as you get a heavy enough inoculum, this works really well. All right, so like I mentioned, one of our staffs is coagulase positive. That only leaves 2 left to differentiate between, and we can do that easily with an antibiotic disc. Okay, so you are going to be, you'll be assigned one organism to do a novobiocin disc test. You're going to take some Mueller-Hinton agar and streak a lawn on there. So remember, if you're using a swab, do that 3 different directions. If you're using a loop instead, which probably you are, since our organisms, I believe, will be on solid media today, So with your loop, maybe you want to go 5 different directions just to make sure you get nice, even, confluent growth on there. Okay. You got the idea there. All right, and then you are, with the forceps, you're going to put your disc. It's going to be an NB disc. That stands for novobiocin. Incidentally, novobiocin is also a DNA gyrase inhibitor, but it actually inhibits it a little bit different way than what the nalidixic acid did. The important thing here is after we incubate it, one of our staffs is totally resistant to novobiocin, and it will grow basically right up to the disc. Actually, resistance is defined as the zone being less than 17 millimeters. So 17 is pretty big, and it's sensitive if it's greater than 17 millimeters. So again, one of our staffs is totally resistant. The other staff has a great big zone around it. After we finish, we'll pretend there's a great big zone around there. Okay. Okay. All right. So you really don't even need to measure this. Usually, big zone, sensitive, growth right up to the disc, resistant. Okay, so when we come back together next time, we'll kind of put together a flow chart, and you can start to see how running these tests helps us identify our organisms. Have a great day.